glass sponge there in the back. That's
Looks like a coral, yeah.
Yep, we gotta get Jess back out.
Yeah, you're good.
For sure.
So even if we rotate the jars again, there's nothing in that one still.
Double check. And let me look up first.
Yeah.
It's an interesting little fish.
Oh yeah. The sun's up. Look at that.
The sun's up. Oh
Looks
yeah.
like it stopped raining.
Nope.
Is it still raining? No.
It's not raining.
Nice wall.
Oh yeah, big boulder.
That's too big. We're looking for a small rock to send up the pipe.
Megan. This is the size of rock you want, Megan, right?
Oh yeah. If we can hug it, we can carry it up.
We've
we
done
have
that.
2 arms.
I know, I've been there.
Yeah.
That's my advisor we're talking about.
Looks like some more permnoids, Acanthogorgia.
How heavy was that last rock?
The carbonate from the last dive?
No, the one that we're talking about hugging.
Oh, the one like with Steve?
Yeah.
That was really big.
I remember we waited in terms of your intern.
I think it was at least a full intern.
Yeah, it was. I thought it was like 0.8 Balkanovs.
Yeah, that sounds about right.
How many lead plates did you draw for that? All of them? Then lead. Sorry,
We
steel.
had already dropped all of our lead weights. We came up heavy.
Wow.
So are you saying you held it in the arms the whole ascent?
Yeah.
Well,
Yeah.
We did that with giant tube worms as well.
Ooh.
Like just carrying a bouquet of like 14-foot long
tube worms, yeah.
tube worms.
really broke
Yeah,
it.
that one, for the tube worms, we came over with the vehicle live and landed with the vehicle live because they were being held in the craft. For the giant rock, we hugged it with Mongo and then laid the craft on top of it, and were able to come over the normal way.
Mango.
Mango. Sorry. Old-fashioned, I guess. Coming up.
Roger.
Holly, did you get some bird shots?
Oh, there's some shrimpies hanging out right there.
You can't really see the island right now. There's some like rain clouds over it. And we're still a little bit far away in order to see the colony. But there were some lesser frigate birds flying overhead, which is really cool.
So Ben, just to add to the image in your mind, we had, I believe it was 2 milk crates mounted onto the front porch with a divider in each one, and those both had rocks. And then we put a rock on top of those rocks because it was a different rock, obviously. You could tell the difference. And then this massive rock, and that was on, so we had 6 rocks on the front porch. And then this massive one went kind of between those top rocks and the forward bio box and got hugged.
So it was essentially a pile.
It was a pile
It was
of
essentially
rocks.
translation deeply traumatic for the data team. The amount of
I like
sampling.
Steve Carey's approach.
No, yeah, the rocks are awesome. It was just like those rocks were covered in biology. We were subsampling every
Right, and we didn't have
every
Dean
single
yet.
thing.
Thank you.
Yeah.
But the other thing that from a Steve Carey point of view was
Sounds awesome.
we were sampling some brittle stars, and it was like a mat of brittle stars. And we used the slurp, and you know, we got a couple, and he's like, you know, I don't know how much biologists really need. Just fill up the bucket.
And meanwhile, the
Meanwhile,
biologist has to put
we don't
each
sleep.
individual one in a separate sample vial or something.
Yeah. Well, that
Yeah,
was
pretty
a
much.
Fun
Yeah,
cruise.
pretty much.
You can't just label it as bag-o brittle stars and go on your way.
It's funny, we were off at the Costa Rica margin recently on the Falcor, and there was these huge swaths of brittle stars, and sometimes you get like an ROV wave packet that would just entrain them all, and they'd just spiral out.
Marley, you want to zoom in center screen there? Marley, can you zoom
Oh,
center
we have
screen?
a-
Yep.
Yeah, let's zoom on.
It's a different one.
Oh.
Yeah.
Shrimp
I'd
City.
call that a point for ROV over science.
Hey,
Way to go, Jess.
Way to go, Jess. But we're a team.
Oh, this is stand by.
Yeah.
Friendly competition.
Yes.
One time we had the Humble watch. We had a Humble off
Looks like a...
to determine who is the most humble.
Oh, Aubrey must have won. Was that that one?
Aubrey won.
Chrysogorgia, but
Yeah.
I don't know.
We gave her a Humboldt squid crown.
Ooh, I want that.
Humboldt squid crown or Humboldt squid? Sorry, this isn't the best zoom for you. I can stabilize and sit down if you want. You want to come full wide there, Marley?
Yeah.
Yeah,
So
you've got some
better
time.
idea.
I'm at like 45 for my camera, so.
Yeah, so I don't keep on hitting a current. Just rather sit down.
Steve
What
Oskovich
was
is on the
that?
chat now. He said, I was on the
Ben,
Humble
what
off
was that?
watch. Did you win, Steve, or did you lose?
Steve was close.
He
to
said I
winning.
wasn't nearly as good as Aubrey.
No.
Oh, she's very humble.
Aubrey is very humble.
But
Alright,
in
go
that
ahead
sense,
and
Steve
Very
did
kind-
win.
kind
That's,
person.
I mean, look how humble he is.
Go ahead, Marley. Let me just do one bump up. Oh,
Yeah.
one minor bump up. My apologies for your eyes. Okay, too far.
That shrimp has another amphipod or copepod on its head.
Does it?
Not this one, but if we look back up.
This one has an interesting purple, like...
Yeah.
Yeah, I can't tell if that's the optics or...
The shrimp?
Would this, could we take a snip of this coral?
Yeah.
There's a little brittle star up on the
Bridge
rock.
nav, you can hold the...
You want to come full wide there, Marley?
And there's no squat lobster on it, which is also pretty interesting. Most chrysogorgids have
Oh, I see what you're talking
Yeah.
a
about.
little squat lobster
It's got a hat.
associate.
Little mustache.
Or
That was a 20-meter step that you had done there, Ben?
yeah.
Okay. I'm going to stabilize myself a bit better here. As I get off the craft.
I'm curious about those scrapes on the rock face. Is that... That's definitely something that happened after it was formed. Because for a second it looks like bird poop.
it does look a lot like bird poop.
But what might abrade those sides?
The striations that you're sort of
Uh-huh.
seeing? Well, there's sediment filling those little kind of gaps, but those could have been partly from the lava being emplaced and breaking through the kind of like glassy quench surface.
Oh, I see.
But zooming would kind of clarify that. I'm not sure if it's just weathering.
Fair enough.
There's a couple of those little urchins with the really long spines too. This one, well, now it's behind the arm, but there's one up there. That little round ballless one.
Oh, yeah. I see it.
All right, Ben, you want to do me a favor?
Do you want to go back?
Oh, you want to stick lock me?
Oh, yeah.
Oh. Yeah, look at that. Not too much.
It's just setting the example, I guess.
Too much jam.
Ben, did we do a step backwards?
No, I just held the current position.
I would do a step backwards.
Okay. I'm going to do just 10 meters.
How much had we moved in that first move?
I think we only moved about maybe
We
8
can
meters.
do 20 meters.
Okay, and
Actually,
then
just from a safe standpoint.
Okay, good. Yeah, because going back in this scenario is safe because we're going down slope for Argus.
All right, let's try this again. All right, someone want to do me a favor?
Yes, got it.
Ready. Guess I'll get this out of the way in case I have to change. All right, Marley, you want to go ahead and zoom in there a bit, please?
This one we can just give a haircut to, probably take like half of it.
Okay. I know this would be a good one to put into the flush, but I don't think we're going to be able to.
I know.
Your heading
Go
is
ahead
shifting.
and come wide there, please, Marley. That's good there.
So are you planning like the starboard bow box then? Not
pull wide
sloping?
there, Marley. Sorry.
Yep, heading
Okay.
shifting.
Yeah. All right.
So when you go into auto head, but you have stick lock on, you're constantly changing.
The
Right,
heading.
so you were, yeah, your auto head was drifting.
Yeah.
Yeah, go ahead.
If you're in auto head before stick lock, then that doesn't drift, but if it's a weird software thing.
Yeah, I forgot to cycle it too. Sometimes if I cycle it, it works all right, but let's try that. All right, we seem stable. Go ahead, Marley, and push on in there again, please. That's good. So we only want a small haircut of this guy.
As
Yeah,
you said, about half.
Probably about
so.
half, because it's not very large.
Okay. Where do we want to put this guy? The flush might not be an option.
Wait, so you want to slurp it or you want to put it in a
No,
bin?
we were just saying,
No.
asking where do you want us
Yeah,
to put
okay,
this guy?
cool. Let's put it in one of the starboard littles if it'll fit. Otherwise,
It'll fit.
yeah, okay, cool. So A, B, or D?
A, B, or D, Roger.
So
about
can you
this
make
here?
out the center spine?
Sorry?
Can you make out the center stem? Because that's what you want to cut, right?
Yeah, they just want a haircut though, right?
Oh, she said about half.
All right, somewhere out here.
I think they want some of the central axis.
Sorry.
So
Sure.
Yeah, so come towards the base a little bit more.
Right here?
Yeah.
You're right behind the bar
Yeah,
and
okay.
bubble, so
Yeah.
you lined up perfectly.
Yeah.
And
I
then
have
just
no visuals.
give it a twist,
Yeah.
and it should.
I think your central axis may not be in the shears. So just work it back and forth a little bit.
Looks like I overestimated where the central part was. You want me to release, and you might lose some bits though.
No, I would just keep working this to see if you can.
Sure.
Shrimp is on for the ride.
I know, I thought he would swim away by now.
No, he's the shape of the shrimp reminds me of other shrimps I've seen on this type of coral.
I don't generally see shrimp on this type of coral.
Ooh, nice.
Nice.
Nice haircut.
This coral reminds me of those fluffy things that you blow into the wind.
Dandelions?
Yeah, I guess dandelions, but
And
there's
like
also other
Like
fluffy.
the
Babies,
bigger
bro.
I was gonna say
Yeah, bigger ones.
it's
Yeah.
it's a bummer that the purple one is stuck in the slurp because this is the perfect
I
slurping
know,
coral.
it really is.
Perfect slurp coral, yeah.
This one actually, well, not a worth the risk, but I was gonna say this one might go past the purple one.
And you said we can't slurp because we're stuck on flush? Is that
No,
what-
we're not stuck
No.
on flesh. We could slurp, where it's just the purple one stuck in the tube somewhere.
Oh, I mean, if,
These are
if we
easily
were on,
identifiable between
yeah, if we
the
were on
two.
bucket 4 and we tried
Yeah.
slurping this, like
We're
we
still on bucket 4.
could, I don't know. I would say it's a reasonably good idea. What do you think, Alexis?
Yeah.
To try to secure the purple one, because I mean we can't slurp anything else then if we get stuck.
Right.
Well, this coral can get stored with the purple one, correct?
Yeah,
Yeah, definitely.
Okay, so affirmative
Just
on slurping.
stack up corals in this flush.
I know, we're just gonna yeah, let's try this one with the slurp, and if nothing comes through, then we know that
We
that
shouldn't
purple
slurp.
one's really stuck.
Yeah, we shouldn't
Yeah.
slurp anymore. Yeah.
Roger.
And if we don't see this one come through, then
We
we're
can
gonna
take
just
a-
go take another snip.
Yeah.
Okay.
Yeah. That sounds
Roger.
like a good plan.
Here's the fun thing, is that looks like this guy's a little bit down here.
Oh.
So 2 and 1.
Give him a nudge.
Nice.
Nice.
Ooh.
Well done.
All right, Marley, you want to do some zoomage there?
That shrimp does look pretty interesting, though.
Zoomage.
Did you see its big
Zoomage.
Which shrimp?
arm? Sorry, the one that's on the coral.
Ah,
I was
yes.
just chatting.
That's great right there, Marley. Thank you.
She didn't think you could hear her.
Well, I mean, I know you could, but
So you're probably going to have to do the side, but
Same
yep,
thing,
you got it.
yeah. On a rock, though, unfortunately, behind me.
Don't.
All right, section is on.
Roger. Looks like we got a good grab there.
Yep,
There we go. Now,
I'd say you're good to go.
I think I'm too butted up against the rock. Between a rock and a hard place.
Since you're, let me
Yeah, I'm too butted up there, I think.
pause that. Spin your wrist roll like 180, and we'll pull it in so that way the Ys are flowing nicely.
Think that way?
Yeah.
I think I just
You're
might
gonna
be
have
too
to
close
wrist
to
up
the slope
again.
here, though. See what I'm saying? Because I'm going to be butted up against that rock.
Yeah, I think you can get it with the wrist up though. Because the distance is the same whether you're going forward or backwards, right?
Yeah.
But if you go backwards, then the Ys are going to pass things instead of getting caught on them. Just a little bit.
All right, ready?
Yep.
Opening up now.
We got a bunch of it.
Nice.
There we go.
All right. Sorry, arm. You're not going to be very happy with me now.
Well, that made it through.
Nice.
Oh, yeah.
There it is. Well, interesting.
Go ahead and come wide there, Marley.
Well done, Jess.
Yeah, that was great.
I'll have to apologize to the arm and give it some tender loving care tonight. Which means I'm going to tighten all of its bolts.
Megan, you see any small rocks you want?
To lodge in the slurp or to collect?
Lodge in the slip.
What are you thinking about doing?
Well, there's actually a little one there. If you zoom in a little bit there, Marley.
Wrong way.
Maybe it might be big rock.
Probably embedded.
Yeah, maybe not.
Hello.
Send a shrimp up the slurp.
I mean, we could. They are everywhere.
Do we want to secure the slurp there?
I would just pumped it on just to see if anything else came through.
Roger. All right, Marley, go ahead and come full wide. All right.
All right, science, are we taking off or are we going for that shrimp? This is entirely your decision.
Oh, the associate, you mean?
No pressure.
I know, but I kind of want the shrimp now. I don't think it would come off if we slurped it, so...
We could force it.
Yeah, there are ways.
You don't think the purple would? Wait, what do you mean?
The shrimp. I
Oh,
don't
off
think
I
the
of
shrimp
think
would...
the shrimp would come
I
off
see.
of the...
I see. Off of the coral.
Do you think it would?
I think it would.
Okay, let's give it a try. Because it is a really interesting shrimp, and we don't normally see that association.
Sure thing.
So we'll stick it in the same.
Yeah, it can go in the same
yeah.
chamber.
We are lined up on the same chamber.
It's gonna
Roger.
get. We'll have to ask the shrimp about the purple coral.
What
we
did
know
you,
that
What
it
did
will.
you see in there?
What did you see in the depths of the slurp tube? Tell us, please.
The depths of the slurp.
Everything.
I need to get this guy first.
I've never been in there.
It's
Probably
dark
not
and narrow.
It's a water slide. It's fun for them.
It's kind of twisty.
It's kind of twisty fun for them. I think we should ask the shrimp when it comes ashore, I think. What do you feel?
I was going to say, let's do it again.
Greetings, shrimp.
The best one of its life and the only one of its life there. All right, Marley, go ahead and zoom on in there, please.
One sec.
That should be good right there.
Data, you're going to want this as 052.
Yeah, I was just considering that.
Okay. You can take your time to decide.
Yeah,
Probably gonna
let's
need
let's
some
do
more zoom
2,
from
5
Marley.
2.
Yeah, I just wanted to see my
Roger.
line up there. All right, Marley, go ahead on pushing in.
There's also that little bit that snipped on the sand of the coral, if you wanted to suction that while we're out here.
All right,
Okay.
so you can
Go
see
ahead,
the
Marley.
shrimp and bubble pretty well. So it gives you a really good perspective.
Yeah, and where's the? Oh, that's the
Mm.
snip you're talking about.
Yeah.
Which one you want first? Maybe the shrimp. 'cause
Yeah,
they'll float
shrimp.
away?
Yeah.
Wait, did you say you want another another snip?
No, no, there's a
No,
little
there's a little bit
bit
that's on the sand that we'll just grab as well.
Okay. I see.
All right, Greg, you ready?
Yep. And watch your bubble.
Ooh.
Wow.
Hey.
Oh,
Where'd it go? Did it go in?
he's underneath. No, he's
You gonna
underneath.
come wide there, Marley?
Oh.
Swimming away.
He just scooted away.
That's okay.
Is that him
Is
there?
that him?
Yep.
That is
Yeah.
him there.
Nav, I reconsidered.
He's, yeah,
No, he's gone.
he learned his lesson.
All right, did you
Shrimp
still want
are
that
hard
little
to
guy
get.
here?
We're good, yeah. We
Okay,
can-
so we're good.
yeah,
Roger.
thanks.
Look at all the other shrimp watching on the ledge.
Yeah, they're like, that can't be me.
They're
They're
like,
like,
"Haha."
nobody move.
If we don't move, they won't
Meerkats.
see us.
Yeah. They're like, swim, Jimmy, swim.
But he did let go. That was the question. No, the amount of water that the slurp moves is rather impressive.
Yeah.
I forget what our 5-gallon bucket test was, like how quickly it
Oh,
emptied a 5-gallon bucket, but it was well under a minute. I think it was closer to 30 seconds.
Wow. That's
Yeah.
very impressive.
it's a lot.
So if I put my hand on it, would it hurt?
Well, it has to be primed and in the water to. It was very difficult to do an on-deck test, actually.
Really?
Yeah.
So
if
like
you
you
suck up
couldn't
air, is it
just
bad
you
for the
couldn't just
motor?
use it as a normal hose on land.
Right.
That's so interesting. It's like how the cameras won't focus unless they're in water.
Well, this is just because the way the pump itself is set up. If it's not primed, it's not gonna, like if it gets an air bubble in it, it's just not gonna work.
Oops. All right, that's all she wrote.
It's so much fun. It's almost numbers time again.
Ben, how far is it to the top of the mound here?
It's about 380 meters.
Cool.
Getting there.
Considering
Getting
that we
there.
did, what, 700 so far?
Look at that little fish.
Yeah.
Yeah, I'm surprised at the progress we've made.
Looks
So-
like a lanternfish.
Oh.
Oh,
The
sorry,
most
there's
abundant
before
fish in
it.
the sea.
Really?
Yeah.
All right,
A McTofu?
ready for you
Or
there,
the-
Ben?
Wow.
Biomass. Yeah.
Oh,
Oh,
in
no
biomass.
kidding.
Huh. I was
They're
gonna
like-
say like in
Uh,
individual count.
No,
I'm
I
sure
think it's in
there's-
biomass.
yeah.
Yeah. And like they're like super crazy in the midwater.
Wow.
Really?
How
But
much
they
of
are
that
also
biomass
getting
is the
overfished.
lantern?
What are?
McTofu.
10%.
10%.
They have all these little like dots on them, so they like glow. I don't know if they bioluminesce or if it's just like reflective dots.
Okay, great.
Yeah,
Safety
they have
gear.
photophores.
Safety gear.
Really?
Do you know about lanternfish?
A little bit.
Oh, yeah.
I think they do have the photophores like along.
Okay,
the,
okay.
Yeah, the ventral side. I'm good, thank you. Yeah.
Anyone?
Actually, I get
Oh, we
confused
have
'cause I
some
think
too.
the bristle mouse, the
Oh,
goddess
okay, never
demandeds,
mind.
are actually
can
the
trade
most
if you
abundant
want.
in terms of biomass.
Yeah, if you guys want.
Oh, really?
Yeah.
Oh, wait, they're the
different
same?
Not
stomiid.
lanternfish.
I don't know.
Yeah.
Okay.
Like a
And when you say
It's a fact.
photophores along the sides, so are those?
ventral side. Yeah.
Yeah. Do they? Are those cells that light up, or are they just reflective?
Yeah. So they're like emitting light. I don't know the mechanism. If it's like an enzyme reaction, like
Yeah.
the luciferin.
But it's actually emitting light, not just reflecting.
Yeah. Yeah, for sure.
Oh.
I'm good, thank you. Wait. What? Deja vu.
Wow. I did not know those were a thing. That's delicious.
The amazement on your face was pretty impressive.
Yeah. Peanut M&Ms are my favorite. I had no idea they made them in dark chocolate. Once again, for those tuning in from home, one of the things that gets us through this is chocolate.
Not a sponsor.
Not any particular brand, but I'm just saying those M&Ms were really good.
Not a sponsor.
Yeah, I think M&M's is a brand.
But we would
Those
be we
small oval chocolate candies with the hard shell where soup's delish.
we
Thank
are
you.
willing to accept sponsors though, if anyone's any
Anyone
coffee
that could dive in.
companies.
Oh my
As a
God.
federal
Can I
employee, I am
please?
not accepting
Sorry.
sponsors.
As a
I
independent
can't
contractor,
speak for the independent contractors.
I
Oh, yeah,
am
as an
accepting.
independent contractor,
Marley,
I
you
can
want to do a
accept
quick swim
sponsors.
there? Just on this nice coral.
Yeah,
Oh,
let's take a
and
nice.
a little glass sponge down there.
Oh yeah.
Oh yeah, that's true. And there's a predatory tunicate next to this guy.
Oh,
That's good there, Marley. Let's have a quick pan over it.
Is it tunicate still
Or maybe
you?
it's, oh sorry, it's just mucus.
Oh.
Oh,
it's interesting though that
it's just
we have
mucus.
mucus.
Sorry, yeah, not just mucus. Mucus is beautiful.
I was
Sorry.
gonna say, yeah.
That is actually really.
Oh,
It is
actually,
weird.
it's super cool.
There are we
Is
don't.
that a mucus, like a mucus sac or something?
Yeah, I don't know what makes them, but sometimes you see them like around rocks and crevices, like all these mucus like nets essentially.
It almost looks like a larvation
Yeah.
house.
It does, yeah.
Huh.
And it does, yeah. Fallen snot house at
Yeah.
the sea.
Does that kind
Quite
of
so.
coral, will that
I don't think it's the
Produce
coral,
mucus,
no.
no?
I mean they produce mucus, but not like that.
not like that? Okay. Oh, and this guy's covered in little
Brittle stars as
too.
well, yeah.
He's just like hanging on by a thread here. Look at that.
Yeah, on that
Oh,
little
man.
ledge.
Wow.
It looks like a spider web.
It
Mucus
does.
net that's holding them there.
Yeah, it definitely looks
Yeah.
like a larvation house to me. Because you see it's kind of like bimodal on the other side.
Sorry,
a
you're
little
gonna
bit.
come a little wide there, Marley.
Yeah.
Apologies.
Can you say what you mean by a larvation house?
Hold on one second.
Okay.
Um,
When you're done navigating.
Yeah, you should apologize, Jess. This current, you know, it's all your fault.
That's a beautiful coral.
Yeah.
yeah, larvations are a phylum Appendicularia. They're actually closely related to the chordates. They're urochordates. So they're more closely related to us than a lot of the inverts we're seeing.
Huh.
And they're these planktonic organisms that you kind of create every day, these
All right,
large
Marley, go
mucus
ahead.
houses that they
I'll
live
Numbers
my
look
inside.
schedule
good.
Here for you.
And
Thanks, Greg.
they
I'm
beat
coming up.
water into that house, so they're
Sure thing.
actively filter feeding the water. And recently, it's becoming more well known how important they are for exporting organic
That's great,
material to
Marley.
depth, because this mucus house is large and it aggregates a lot of other organic material as it falls and gets heavier. So they're neat components of the plankton.
Oh, wow. That's
Mhm.
really interesting.
Yeah. Yeah, and they have the.
Yeah, look, it's just like caught on there, so.
I know.
Fishing net of mucus there.
It also reminds me of those giant bubbles, you know?
And
that
I'm
you can-
almost looking full down.
Roger. All right, you're gonna come full wide there, Marley.
Do you
I
want
think
me to
this
hold
is a
current
type of
position?
Narella.
That's okay.
That's
we'll,
my
we'll hop
We're
off
not
right now.
sampling. Okay.
guess for
No.
genus on that coral.
Off we go.
Is that like a coincidental growth of that kind of mucus sac there, or
It's likely deposited from the water
Yeah,
column,
it was
so
just-
it sinks and then serendipitously got caught on that ledge. We'll have to leave a tissue.
Look at that map. It's a nice map.
That is a gorgeous map.
The Maltese?
Call that like a tropical colored map or
Oh
something.
yeah,
I don't
the
know.
color
Man.
What
scale
would you
is
say?
really
I
on point.
love a good map.
The trick is to not oversaturate.
It reminds me of the color palette of my grandmother's living room.
Right there with you.
Yeah, right?
Right there in your grandma's room.
Yeah. I can see it.
Trying to get that purple one out there still.
Yeah, did you watch your pressures drop?
No, I saw
Oh, okay.
the coral move there.
Yeah, the slurp is a pretty big flow hog. So if you watch your pressures, right, you're at 2.5.
Yeah.
That's an anemone.
Oh, yeah.
Oh, well.
1988.
Couple more corals.
Coral there on the left.
Mm-hmm.
We haven't really talked about this dive, Alexis, but like, why are you interested in corals at this depth? Like, what are the things that you're looking for and in your
More
research?
our
Oh,
lab
wait,
is-
wait, wait. Before you answer that,
Yeah.
which came first?
Uh-oh.
Deep sea fascination or coral fascination?
Oh, that's an interesting.
I think deep sea fascination for me.
And then corals, okay.
Well, so I became really interested in animals that can live like over really large depth ranges, and corals are one of those. So I just started mostly studying octocorals and like the chemical makeup of their skeletons, and then when I got more into octocoral research, I started studying deep sea corals. And then I started wanting to do more live work, because what I was doing during my master's was museum-based, so I was using preserved specimens, which is great, and natural history museums are essentially treasure troves for lots of types of research, but I wanted to do more of this kind of real-time live exploration and study how we are currently impacting corals, so that's where my interests, how my interests developed. But for what Holly was asking, we're really, our lab is looking at the distribution of corals throughout the Pacific, so across seamounts, from south of here in the Phoenix Islands protected area up through Johnston Atoll and Palmyra and Kingman Reef, and looking at like where are corals distributed. Are they distributed differently across different depths? Do you have different
zoom in here a
species,
little bit, Marley,
or are
please?
you seeing more homogeneous communities?
Marley, you want to zoom in a little bit on the center screen?
Yeah, let's take a look at this. Looks like this one might have fallen over, which could be a good candidate for collection.
Marley, you want to go ahead and zoom in on the
It's
I love
just the little
collecting-
inflection
Collection.
at the end, giving me the vibe of when your mood
Work
is.
it.
Oh my gosh, yes. I would love to collect this.
Yeah.
Yes, please.
Yes, you
love
would.
it.
Yas.
Yeah.
All right, go up.
Get
on
it.
the shift there, babe. You're gonna come wide there, Marley.
So
I'll
I
get us
try,
positioned.
well, I try and take advantage of like, of
And we'll
natural,
probably do one step back a little bit.
when
It's
nature
already dying.
has already knocked it over.
Yeah.
So likely that coral would
You're gonna
not
take it back one.
continue to grow over time. So sampling it is not necessarily being as destructive as if it were. And these ones can be relatively fragile, so their skeletons are really hard. So when we grab it with a manipulator, it'll break in a couple places, and so if it's already fallen over, then we avoid breaking a really large living colony. And that's a great specimen for age dating as well. Like we did with a bamboo coral on a different dive, we could section the base of that coral and it's still alive, so we know we can get an age for it, and know that it was still growing.
So, and what are the ways, how would you age that coral?
You can use isotopes, so either radiocarbon dating or uranium thorium, and essentially it's just ratios of isotopes changing is how you measure that, and both those two different types can measure different, essentially different levels. Like, so carbon dating can do more recent times, and then uranium thorium you can use for older corals, or older skeletons that might be like thousands of years or hundreds of thousands of years.
Some of these corals are thousands of years old, right?
Yeah, the black corals, which we've seen one on this dive, those ones can be really, really old. A single colony can continue to be alive for over thousands of years, and these other corals are often within the hundreds of year ranges, just depending on the size. And they're just because they grow very slowly, so they don't look super, super giant, but they just have been growing for a really long time.
Are you able to keep track of like changes in growth rate? Is that data you're able to?
It depends on the coral. I think for some I don't do it personally, but for some bamboo corals, they do grow in like tree ring formation, so you can see the sometimes annual rings, and it looks, and some people can, when you do really detailed age dating, you can date each ring, and so you would be able to see how quickly that was growing, and
Oh, wow.
and if certain rings are thicker than others, then those would be growing probably more during that frame versus like a really thin ring.
How far do you want me to zoom?
Nice Argus.
Yeah.
What kind of coral do you think this is?
Anelopsamia.
Nelopsomnia, okay.
Oh, I think we collected one earlier in the dive
Yeah.
too, but because this one's made itself available to us.
Yeah. I think it's just so interesting what you were talking about. You guys are looking at distribution of corals across all of these different, you know, Kingman, Howland, Johnston, Palmyra. These are all protected places that right now we don't have a lot of baseline information about. We don't know what's growing there or in what distributions or at what depths. And that's information we need.
Yeah.
Yeah.
And it's interesting to think about, like, these span really large geographic areas, right? So if you have similar coral distributions, thinking about the connectivity of the seamounts and how they provide these essentially like oases in really vast ocean space for these corals to grow.
I mean, it's interesting that you say that. So yes, for corals to grow, and then even terrestrial species as well. Like we look about how species migrate to these island colonies, and these atolls are sort of stepping stones.
Right, and it's exactly the same under the ocean. People think that seamounts provide stepping stones for a lot of species to kind of expand over these large areas. Oh, that's beautiful.
Wow.
Nice, good pick.
Nice
Nice.
job.
Such a delicate touch.
That is a very delicate grip there.
I can't hear
Yeah,
you.
Jess, I can't hear you.
Sorry, I was off SPL. You
No
want to go
worries.
ahead and zoom in on the sample real quick?
Yeah, so you can see the little pink pockets in the
Yeah.
polyps. That's the live tissue.
Yeah.
And it's actually covering most of that skeleton. It's just a really thin layer.
Wow.
All right. Do you guys have your images that you got there? Megan?
Yes. Very happy.
Go ahead and come wide there, please.
And this is also a really good example of how you get coral rubble. So we've seen it on some of the other dives, and we'll likely see it as we get more shallow. So these corals are growing, and eventually it's either generally that they become too heavy, and so they fall and they break. And so those pieces will die. Sometimes they'll continue to grow, but largely in an area like this, they'll just die. But they create all this rubble underneath, and that we think that that's been happening. I mean, we know in other areas because we've dated them that that happens for hundreds of thousands of years, and so it just continues to accumulate and can provide the coral rubble itself. Actually, can have a higher density of invertebrates than the live coral, so it creates another type of habitat for another suite of organisms.
That was another
Nice.
question I had
Good call.
actually as you were talking with Holly before. You were mentioning the kind of coral density geographically, but we're about to crack 1,000 meters here.
Yeah, that
Is
was a
there-
good move.
Is there a distribution by
Thanks for
depth
showing
that
me
we're
a bubble,
expecting
though.
to see?
Yeah, I don't necessarily know what we're expecting here, but that is a pattern that's kind of continuously observed with different like species or genus of corals, in that they will inhabit a specific depth range. And like, so Caligorgia is a good example, one of the primnoids. You have very specific species at specific depths. And so what we can do with this video in the physical samples when we get back is really tease apart the different species that we're seeing, and try and observe if we're seeing that same type of pattern of specific ones at specific depths across these seamounts or islands.
Do you expect this to be very floaty?
No, it will be
No.
heavy.
It's surprising to me the consistency you see in the depth range when like across like a broad geographic range. When I imagine that more like other environmental conditions like
I
oxygen,
feel like you should put black tape along the
maybe
tops.
I would
should,
expect that to
yeah.
be more influential.
Yeah,
you know,
yeah,
for them to be so fixed at depth.
yeah. And some of it is
So you're
temperature,
pretty solidly over C.
because temperature can be a big
But we're going
kind of
you're going
barrier
D.
for certain
Yeah, just to
species.
see if we can get
Oxygen too.
But I'm sure those participate in your model.
Yeah,
Yeah.
yeah.
Oh, you're on the edges of the fingers.
That's cool. I never thought about
It's hard
that.
to
Like
see this one.
the distribution of coral in the, we'll call it in the y-axis versus the x-axis.
Yeah.
That looks pretty good.
Yeah.
I would let go. I'd be inclined to let go here.
Yeah.
There you go.
Great.
Nice. Really well done.
You're caught
Yeah,
on that
for
back.
Talked about
Yeah.
these seamounts as being stepping stones for marine life. When they're islands and atolls, they're stepping stones for terrestrial life, so birds and you know, all kinds of species that have migrated across the islands. But they're also stepping stones for humans. So Howland and Baker and most of these Pacific remote islands have evidence not just of like historic use in terms of, you know, the whalers and the guano miners in World War II, but evidence of ancient Polynesian seafarers actually using them as well and stopping by on them. So we find evidence in the terrestrial species and also just archaeological evidence that ancient Polynesian seafarers were using these islands.
Wow.
Yeah.
That's really cool.
That I froze
And it
up.
all starts deep in the mantle.
Our
The
resident,
volcanologist
yeah, our
brings it back
resident
home.
volcanologist.
Lest we forget.
It does though. Geology is the basis of life.
Life-bearing rocks.
Life will find a way.
Yeah. Classic line.
Grandma M is speaking to Megan and says she's very interested to learn more about our life-bearing rocks.
Hi, Grandma.
Well, Holly, I'm sure you have, and anybody else familiar with Hawaii, the ancient Hawaiians believed that every single thing in nature has a life force called mana. So not just plants and animals, but rocks and soil and everything has a life force. Have you heard about that, Holly?
Yeah, I have.
Yeah,
and
let's do
I
it.
probably wouldn't try to get too deep into like, you know, Polynesian cultural understandings, but yeah, mana is a life force that is acknowledged definitely in Hawaiian religious beliefs as well.
I just think that's a nice idea.
No, it's super cool.
I like
And
rocks.
Yeah. Yep. Absolutely. No, you're totally absolutely right.
Kidding aside, right, this is the leading theory for how life formed on this planet. We, in school, you know, people my age, we learned about like Darwinian pools, but this is not probably the most scientifically accurate. Instead, it was this kind of like interface of acidic and basic conditions at ocean floors where, you know, you get this kind of what's called chemosynthesis, right, in between the ocean floor and acidic water, back when there was more CO2 in the ocean. So really, without geology, we might not have ever had
Oh, totally.
RNA.
Yeah, I mean, yes. Most of the nutrients, I think, in so we think of photosynthesis, which is a huge part of primary life, but chemosynthesis is likely just as significant, if not more significant, to the overall primary production, and that's fueled by essentially geologic forces. So I totally think what you're saying, Brandon, is on par.
Yeah, we just see photosynthesis. You know,
Right,
all
we're
around
more
us.
familiar
It's
with it.
terrestrial.
It's a beautiful coral.
Shall we look while we have a time?
Sure.
Okay, I'll stabilize myself here a little
It's
bit.
got a number of friends.
All right, go ahead, Marley.
I'm interested at what's at the base of that. Looks like
Yeah.
Madrepora, the other type of scleractinian hard coral.
Oh, the Z.
Yeah, the
Yep.
zigzags.
Oh.
Oh, a homolid again. Look, he's
Yeah.
got the same type.
Cool.
Oh, and
Yellow
another one
is
up
in.
there.
Yep, 2 homolids.
Yellow is
one
in this
kinda
season.
But that one has a
Yellow is
different.
it?
The other homolid has a different type on its back. See the little it has like a little pink puff.
Oh yeah, it does.
Well, that's why he's not front and center.
Too avant-garde.
Too avant-garde.
Yeah, that's definitely
Maybe
a,
he's bringing in the new fad. You never know.
Yeah.
a
It's always
clump
gotta be
of
one.
Madrepora back there, which is cool to see. We haven't seen much live. And this other coral looks like it's really just kind of settled on top of that. I'm curious who settled first.
That crinoid is waving at us. I don't
It
know if you
is.
guys know that or not, or saw that. It's actually waving.
Yeah, we go way back.
So are we seeing, do you guys see the rippliness?
Yeah,
Yeah.
the shimmer in
Yeah.
the water.
So we have probably 2 different water masses?
Yeah, with a little thermocline or something.
Yeah.
Ooh,
Whoa.
that's cool.
Yeah, that is
Little
cool.
front over here,
Do we have a temperature
Right.
reading on Herc and a temperature reading on Argus by chance?
Let's see. Temperature reading on Herc
It's 4.7.
is
All right, Marley, you wanna go ahead
4.7,
and
but
come my way,
Argus
please?
does not have a water
Oh.
temperature reading.
No.
Okay.
Marley, you want to go ahead and come my yeah,
That's
thank
super
you.
cool though. I love seeing that.
Yeah, just a couple minutes ago we were a little bit cooler, but not super significant.
True, we'll see what we are when we keep going up though.
Yeah.
Yeah.
That kind of ripply effect reminds me of when you can actually see the horizon, you know?
Mm-hmm.
Yeah.
It's really
Oh,
cool.
yeah. Like a
Yeah.
mirage or
Indeed.
Yeah, it's
There was
refraction
on the
from
last cruise I
density
was on in the
gradient.
Atlantic, we found a spot that was like essentially the intersection of 2 water masses, and D2, the ROV on Okeanos, was reading 4 degrees cooler than the vehicle that's analogous to Argus.
Oh wow.
Sirius.
Sirius, yeah. So there was
I feel
definitely.
like they started off with Sirius and then like lost their naming convention.
Yeah.
But
They became casual.
because
another rock
Sirius is
no. Cool.
the dog star.
Dog star?
Yeah.
What is that one?
Like astronomical.
Yeah, there's like Argus is the monster with all the eyes that
Right.
sees everything. And then I forget the mythology associated with the name Sirius, but it was like, you know, that which lights up, you know. So it was again
Similar concept.
mythology, and then it was like D2.
And then it was Deep Discover. Yeah.
I was like,
No.
It's okay. People can do what they want.
Naming
yeah.
conventions can be
I just feel like, you know,
powerful.
with Deep Discoverer, you could have had like Robot Rising as instead, and then you could have
Robot
R2D2.
rising.
Ooh.
It's a terrible joke, but I've been saving
Oh,
it
what's
for
that
years.
pink?
That's great.
Can we take a
Now
look?
it's your
I think it's a sea
Oh.
star, but it's
Yeah.
definitely different.
Sure thing.
Is it a cookie?
I don't know.
Just a little bit burnt.
That's considered a raspberry tart, actually.
Yeah. Oh, a couple black corals right here, like we've seen.
All right, Marley, you want to do a real quick zoom in?
Wow, that star is super cool. I've never seen
It
something.
is
Oh, it's
not
an
a star.
urchin.
Oh.
Oh.
Hey, urchin.
Indeed.
Ooh,
I want to pop down here a little bit.
Nice camouflage.
yes.
Really
That's
blends
super.
into
I've never
the
seen
pink
like
rock.
one with that
star
radial. Yeah.
right?
Yeah.
It's like pentaradial.
Yeah, which is like
coloration.
essentially their
Their
their radial
Yeah,
symmetry is, but that's really
Radiant.
cool.
All right,
Radiant.
Marley, go ahead.
Radical.
So that's
Radically
considered a
radial.
spicy
Say zoom
raspberry
in or zoom
tart
out?
because of the
Zoom out,
spikes.
yeah.
Okay,
Yeah,
sorry, I couldn't
very
hear you.
spicy.
It's all right. We're getting hungry. I think we're talking
I know.
about raspberry
Yeah.
tart.
Is that a
or-
raspberry?
Is it breakfast
time?
We are 30 minutes from breakfast right now.
Is there a cup of coffee down here I can grab?
That was a good sigh there, Marley.
Everything's fine. No, I'm fine. I'm fine.
Some black corals here too.
Yeah,
Okay.
there's getting to be a couple more of them. Looks very similar, probably the same ones we saw yesterday.
I think it's probably a different one than we saw yesterday.
Why?
They don't
Oh.
migrate that far. Not in this
Okay.
time scale. Forever. There's an orca
Getting
with
sassy up there.
over
Oh yeah,
Yeah.
here we go. Some rubble.
Oh.
Fish.
Is that an Oreo fish?
It is an
It is
Oreo
an
fish.
two Oreo
Two
fish.
Oreo
Ah,
fish.
we
You
have
could
a
make
sandwich.
So-
you could make a full Oreo.
I think that this is a very like
Oh,
broad, not scientific. Haven't actually like
This
done
might-
any
Okay,
analysis,
this
but
might be a good spot for a rock sample.
ooh,
Actually,
Yeah, let's
Oh my
totally do it.
there's definitely
Get excited.
some correlation to when we see Oreo fish and when we see high density corals.
Huh. Yeah.
This is a really cool.
yeah.
Yeah, look
Ooh,
at
I would
these.
wow.
This is
step back
this is
though.
nice. Yeah.
Oh, this
Ooh, yeah.
Look at the size of those crinoids. They're so big.
Yeah, this is like so.
You
This
can
is
zoom in a little
getting
bit
on
there if
the
you want, Marty.
order of vertical reef.
Well, we'll let me start
Yeah.
to move forward a little
Oh,
bit because right
yeah.
now
But
you're
see
at tether
how-
extent.
And this
There's-
is where I want to rock. Oh my gosh.
So we
Yeah.
don't
What a
play
good
it
end
off.
of dive.
This is super nice though. It looks like actually, let's take some zooms before I.
Shrimp.
Wow, this guy's cool.
Look, there's a lot of reef rubble
Look at
over
that
here.
Yeah.
spot lobster in there hiding.
Look
This
at
is
all that
awesome.
Rubble.
If we
This
could
is
take
like
a
the
zoom
rubble
on the
field.
live stuff
Sure.
right here.
All right, Marley, go on
This is
ahead.
fun.
It is. Exploring the ocean is fun, Alexis.
So this is nice.
It's like the first time Alexis
That's
said
great,
that.
Marley.
This is really fun.
Look at all those guys hiding in there.
Wow, look at all of that.
Yeah,
great,
so we've got
yeah.
some squat lobsters, different types of squat lobsters in there. That looks like madrepora, so a nice, but it's interesting because it's so brittle that it just gets heavy and it starts to, I think, break
Fall over.
off. Yeah, which is where we get the dead parts. But it looks like both madrepora and anelopsamia maybe have been growing here,
I
which
can
is
come up a little bit. if you want.
interesting. There's a little chubby sea star up there. He's been feeding.
Oh, does that looks like a cookie
Something.
system?
Maybe.
And
Looks
not
like
just
a cookie.
not just because
I believe that was a biscuit.
A
or
biscuit.
a ravioli sea star. That was a ravioli one.
If we have the time to kind of go around this rock, like look
Yeah.
at the other side too.
Yeah, we stopped the ship because Megan was talking about a rock sample
Cool.
too.
Yeah.
So.
And we can look for a rock sample.
Wow, there's just so much
Jessica
going on.
pirouette.
And so that's
Yeah,
like where
yeah.
you.
Current.
This is again a good
Challenge
example of
accepted.
the
Yeah, exactly.
how biodiversity is enhanced by these coral habitats.
Yeah.
So you have a lot more organisms that are aggregating on the coral than if you just have the bare
Yeah.
rock.
Yeah.
This would be a nice coal map candidate,
Oh, yes.
right?
Yes, it's perfect.
It's a
actually.
perfect coal map candidate.
What's a coal
Wait,
map?
what is it?
Coal map? It's like a 3D reconstruction
Yes.
pipeline. You just would take like a bunch of images around this, and
Oh.
it's like a perfect feature.
This is
You
awesome.
can get 3D
Yeah.
from a monocular camera from the
Yeah.
structure
Oh, wow.
of the
Yeah.
motion of the camera.
Yes.
Yeah, it's actually what I was thinking about was I was going to ask Ben if you were out here the last time Chris did that.
Oh yeah.
Yeah.
When he was clicking on that. It's become a new
It's a new
favorite
fad.
tool, and now Megan's into it.
I'm
Wait,
into it.
it's super neat.
Oh, it's so, and it's just open source and ready, and
It's awesome.
it's robust. Yeah,
It's really cool. I've been coal mapping random things on the ship.
Sure. So, the cleavage of this
Yeah.
rock is quite different.
Yeah, I know. It's fractured in a pretty unusual way, but
Uh-huh.
I mean, this whole thing looks like a large boulder that kind of maybe rolled down.
from the
Yeah,
top, but we're still pretty close to the top.
I know. So I'm curious, like, it looks like we have some tighter spaced contours that we're approaching maybe, so there might be a little bit of a steeper slope and then
Yeah.
something flatter, but yeah, I don't totally know where this kind of erratic basalt block came from, but definitely has a different
This is awesome.